Now we discussed about PCR, that is polymerase chain reaction, different steps of polymerase chain reaction and some other terms like biosafety, biopiracy, biopatent, DNA fingerprinting, genomics, genetic counseling and pedigree analysis. What is the importance of PCR, polymerase chain reaction? PCR helpful for preparing much amount of DNA parts from a little sample. This technique is useful in biotechnology and DNA fingerprinting. Now we discussed about the steps of PCR. The steps of PCR, that is polymerase chain reaction. PCR, DNA polymerase enzyme. There are different types of enzymes are used in PCR. DNA polymerase enzyme is used repeatedly for making many copies of a small fragment of DNA. So named PCR. DNA polymerase enzyme DNA polymerase enzyme is used repeatedly for making many copies of a small fragment of DNA, so named PCR. Then steps of PCR. First, double helical DNA molecule. We already discussed about the structure and double helical structure of DNA in previous sessions. You can utilize it from our channel. The double helical DNA molecule is heated so that it breaks up into two strands. First step, double helical DNA molecule is heated so that it breaks the double helical structure of DNA. The double strand of DNA breaks up into two strands. Then next step, primers are added and the DNA is cooled. Primers are added and the DNA, heated DNA is cooled. Next step, DNA polymerase enzyme is added and in its presence, the presence of this enzyme, the two single-stranded acquire complementary strands and so two molecules of DNA are formed. DNA polymerase enzyme is added and in its presence, the presence of enzyme and in its presence, the two single strands, the double strand is detached from each other and form single strands in first step. Then this single strands acquire complementary strands. That is the photocopy of primary strand, that is a complementary strand. occur or prepare the complementary strands. And so, two molecules of DNA are formed. These steps are repeated. First, double helical DNA, that is a natural form of DNA molecule, is heated so that it breaks up into two single strands. Primers are added and then cooled. The DNA polymerase enzyme, then add DNA polymerase enzyme, and in its presence of DNA polymerase, the two single strands acquire complementary strands or photocopies, and so two molecules of DNA are formed. These three steps are repeated to get multiple copies of DNA. These days, DNA polymerase from a bacterium living in hot springs. Where from we get DNA polymerase? The DNA polymerase enzyme get from bacterium, one of the bacterial species living in hot springs called TUG polymerase is used in PCR machines. The name of bacterium that produce the polymerase enzyme is TUG polymerase. This is used in PCR machine. Now we discuss main three steps of PCR. Then next term, biosafety. Biosafety. What is biosafety? Department of Biotechnology, in compliance with Rules of Environment Protection Act, Rule of Environment Protection Act, EPA, give permission for research and use of genetically modified GM organisms only after testing its safety to humans and other animals and the environment. That is known as biosafety. According to Environment Protection Act. Environment Protection Act, EPA, give permission for research and use of genetically modified, that means GM, genetically modified organisms, plants and animals, organisms only after testing its safety to human and other animals and environment also. This is known as biosafety. Next term, biopiracy. Biopiracy. What is biopiracy? That means, piracy means theft. Biopiracy means patenting or exploiting a biological resource of another country without being authorized or giving adequate compensation is known as biopiracy. Biopiracy means patenting or exploiting a biological resource of another country without being authorization or giving adequate compensation, known as biopiracy. Sometimes a useful biomolecule extracted from a plant growing in another country may be patented and used for commercial benefit. Sometimes a useful biomolecule extracted from a plant growing in another country may be patented and used for commercial benefits also. That is also included in the terms of biopiracy. Example, US granted a patent for germplasm of basmati rice grown in India. Example of such germplasm biopiracy: the US granted patent for the germplasm of basmati rice grown in India. Biopiracy means patenting or exploiting a biological resource of another country without being authorized or giving adequate compensation. That is known as biopiracy. Sometimes a useful biomolecule extracted from a plant growing in another country may be patented and used for commercial benefits. Example, US granted the patent for the germplasm of basmati rice grown in India. Next term, biopatent. Biopatent. What is biopatent? A patent is an official document. A patent is an official document. Possession of this document permits the handler to use or sell his or her invention. That is known as biopatent. A patent is an official document possession of this document permits the document holder to use or sell his or her invention. That is known as biopatent. In India, duration of patent is 14 years. In India, duration of patent is 14 years except for food and pharmaceuticals, which is only for 7 years. In case of food and pharmaceuticals, the duration of patent is 7 years. In other cases, it is 14 years in case of India. A patent is granted for 1. An invention or discovery. The patent is granted for an invention or discovery or improvement of an earlier invention. Patent is also given for improvement of an earlier invention or process of generating a patent, a concept or design. For mainly there are 4 cases patents are given. First, an invention or discovery. Second, improvement of an earlier invention. Third, process of generating a patent. Fourth, a concept or design. Next, DNA fingerprinting. DNA fingerprinting. Next term. DNA fingerprinting. What is the importance of DNA fingerprinting? A DNA sequence of each person. The importance? A DNA sequence of each person are unique. That is never multiplies. The DNA sequence of each and every person are unique. DNA fingerprinting used for murder cases, rape cases and for find out who is the actual father of child. The importance, the point that noted is the DNA sequence of each and every person are unique. DNA fingerprinting used for murder case, rape cases and find out who is the actual father of a child. Then steps of DNA fingerprinting. Steps of DNA fingerprinting. DNA is isolated from blood or semen. DNA is isolated from blood or semen. Blood or semen, etc. Its quantity is increased by PCR. We already discussed about PCR. Its quantity, DNA quantity is increased through PCR technology. Then the third step, the length of DNA pieces vary from person to person. That is very important. Then identify the length. Identify the length of DNA. The length of DNA pieces vary from person to person. So detect the length of the DNA. That is the third step. Then fourth, DNA pieces separated from each other according to size. DNA pieces separated from each other according to size and change with the help of a technique called electrophoresis. Fourth, DNA pieces separated from each other according to size and shape with the help of a technique called electrophoresis. There are main steps of fingerprinting. DNA is isolated from blood or semen. Its quantity, the DNA quantities are increased through the technology PCR. The length of DNA pieces vary from person to person. Identify the DNA, then DNA pieces separated from each other according to size and shape with the help of a technique called electrophoresis. Then next term we needs to discuss genomics, genetic counseling and pedigree analysis. Next, genomics. What is genomics? Next term, genomics. Genomics. Genomics is a collective term for a full set of genes in an organism. Genomics is a collective term for a full set of genes that denoted in DNAs. DNAs are the part of chromosome. Genomics is a collective term for the full set of genes in an organism. So, genome means all the genes present in a haploid set of chromosome. Genome means all the genes present in a haploid half number set of chromosome. What is the importance of, what is the point that denoted the haploid set? Each gene with a pair, that is allele. Genes are in pair condition, that is allele. So only we need to check half number of chromosome, that is the haploid. So we get the next the allele with the identical gene. So we only need to check the genes in a haploid set. Genomics is a collective term for a full set of genes in an organism. Genes in an organism. So genome means, so genome means all the genes present in a, all the genes present in a haploid set. Haploid set, that is half number. Genes are in pairing. One is identical to another in an allele. So we only need to check the haploid set. The genome of E. coli bacterium, yeast, that means Saccharomyces and Drosophila are already known. The genome project about E. coli, yeast, that means Saccharomyces and Drosophila are already known. The human genome mapped in 2003. The human genome mapped that start in 2003. Humans have 23 pair of genome or chromosome. Humans have 23 pair of chromosome, that is 2 and 46, and a haploid set, that means 23. Human genome has 3 into 10 raised to 9 nucleotide base pairs. The genome of E. coli, yeast, that is Saccharomyces and Drosophila, etc. are already known. The human genome mapped in 2003. Human has 23 pair of chromosome. We already know the diploid set. 2n number is equal to, in case of human being, the 2n diploid set is 46 and haploid set is 23 chromosome. Human genome has 3 into 10 raised to 9 nucleotide base pairs. Nucleotide base pairs. We discussed about this part in another session in our channel. You can utilize it. Next term, genetic counseling. Genetic counseling. What is genetic counseling? What is the importance of genetic counseling? This means advice given regarding a genetic disorder so that the couple knows whether to give any more children if their first child is suffering from a genetic disorder. It is the advice or information for couples who are already suffering from a genetically disordered children or child. The genetic counseling means advice given regarding a genetic disorder. There are different types of genetic disorders we already discussed in other sections of our channel. There are different types of sexual and autosomal disorders. We know it. The genetic counseling means advice given regarding a genetic disorder so that the couples of family, the couples knows whether to have any more children if their first child is suffering from a genetic disorder. That is the importance of genetic counseling. Genetic counseling means advice given regarding a genetic disorder so that the couple knows whether to have any more children if their first child is suffering from a genetic disorder. Last part, pedigree analysis. Last point, pedigree analysis. What is the importance of pedigree analysis? The pattern of inheritance of a particular trait. Trait means feature or character. The pattern of inheritance of a particular trait. Inheritance means the characters given from parents to next generation, that is inheritance. The simple meaning of inheritance. We already discussed detail about the genetics in our channel. You can utilize. It is a long session up to hours, taking hours. Detailed pictorial representation with notes for examination. The pattern of inheritance of a particular trait or feature among humans is identified by the method of pedigree analysis. The pattern of inheritance of a particular trait or feature among humans is identified by the method of pedigree analysis. It is the diagrammatic representation of pedigree analysis is a diagrammatic representation of relationships showing a particular trait in a family. It is the diagrammatic representation of relationships showing a particular trait in a family. Trait in a family. These traits in case may be a genetic disorder. We can consider trait means genetic disorder. It is the pedigree analysis is the diagrammatic representation of relationship showing a particular trait in a family. Example of a pedigree chart of autosomal disorder. We can draw pedigree chart of a autosomal disorder. Example, a pedigree chart of an autosomal disorder. There are different types of autosomal disorders and sex-linked disorders. You can utilize our channel and can learn about these autosomal and sex-linked disorders in detail. Now we represent an autosomal disorder, the pedigree chart of an autosomal disorder. Here circles are females, squares are males, filled circles and filled squares are affected males and females. Empty circles and squares normal case. Here circles are females and squares are males. Filled circles means affected females and filled squares means affected males. Then empty circles normal females and empty squares means normal males. Circles are females, squares are males, filled circles are affected females, filled squares are affected males, female circles are affected females, and empty circles are normal females and empty squares are normal males. Now we complete this part. If you like this class, you can like, share and subscribe our channel for further classes.